Dr. Bimal Singh, please continue.
Good afternoon, everybody. So, it is my pleasure to introduce today's first, that's Prince Yadav. U.S. Yadav is a principal scientist at XOD, Division of Physiological Production, ICR, CRG Star. Dr. Yadav has such experience for 42 years, including 60 years as principal scientist. Under his leadership, CRG became Central Institute of Cloning that would name the Guitar Gaurav. Recloning of cloning of Apanis, Buffalo Good Field, Matthew clone of Buffalo for which his name is recorded in India's Book of Records. In addition, Dr. Yadav and his team has produced 30,000 frozen human doses from cloned wood and more than 100 proteins through AI from the cloned wood. Dr. Yadav has working experience in vitro fertilization, over pickup, plant genetics, genome editing, livestock reproduction technology with reputed laboratories in Germany and China. He is the recipient of several prestigious awards, including Team Leader, Nanaji Deshmukh ICR Award, Distinguished Scientist, Indian Society for Social Development, National Fellow, National Academy of Daily Sciences in India, among others. He has guided 10 students, including 12 PhD students, and authored 4 books, several book chapters, and edited books. So I invite Dr. Yadav to present his lecture. Dr. Yadav, please.
Thank you very much for the introduction. All are listening. My voice is coming to you.
Yes, sir, it is audible.
It is okay. So may I start?
Yes, sir, it is.
Okay. So I will talk to you for next few minutes on animal cloning, status and scope in Indian livestock production. So first thing is animal cloning. Animal cloning is a asexual means of reproduction. That means the normal male and female breeding is not required for this process. And this genetic copy of the donor animal is created. It is similar to the identical twins. In identical twins, by a natural process, in some rare cases, the embryo splits in the womb itself and makes two individuals, and both are similar. Twins can be identical and non-identical. If these are from the different sperm and eggs, they are different. But if they are from a single embryo splitting, then these are identical. And clones have predetermined sex. Reason? Because we are using the somatic cell which has 2n number of chromosomes. And these chromosomes are coming from a single animal because we are not using the sperm and the nucleus of the oocyte is also removed. So only the genetic information which is passed in clones is from the donor cell. So normally there are two methods of cloning. One is through the micro manipulator, which is a machine attached with the microscope. You can play with the oocyte and embryo. And another is handmade, that is micro manipulator free, in it which the main process is manual through a blade. We cut the oocyte with the help of a small microblade to remove its nucleus. And the matured oocyte cytoplasm is attached with the donor cell. So you can have a look on the process. Those who are understanding the biology of reproduction, they can see that there are two things required to start the process. One is the animal which we want to clone. It can be male or female. From this animal, we collect the small tissue, very small teeny tissue, and from that the primary somatic cell culture is established. In this photo, you can see that the dark part is the tissue and rest the cells have grown out from the tissue. From these cells, after confluency, we either freeze them or use one cell for making a cloned embryo using the cell as a donor cell. On the other side, we have ovaries. From ovaries, we aspirate the follicles and we get the immature oocytes. These oocytes are matured in the culture media and then in micro manipulation free cloning or handmade cloning, the zona is removed, and when we remove the zona, there is a protrusion cone which tells us that the nucleus is on this side, and we cut it with a micro blade and discard the nucleus part. About 30 to 35% of the mature cytoplasm is removed, which is having the nucleus. The rest of the mature cytoplasm, we take two such cytoplasms and one somatic cell and fuse with a machine known as electrofusion machine. Then the embryo starts growing in 2-cell, 4-cell, 8-cell, morula and blastocyst stage. Blastocyst stage reaches about 7 to 8 days. Then this blastocyst is transferred in the animals. These animals are synchronized animals and they were in heat 7 to 8 days prior to the transfer, because the age of the blastocyst is 8 days. So animals should also be in the cyclicity at the same stage. So we palpate the corpus luteum, check through the ultrasonography, and then transfer the blastocyst in the animals deep in the horn. Then these gets implanted, and at 28 days we check the pregnancy. If there is such dark vacuole, then animal is confirmed pregnant. And after completion of gestation of about 310 days, calf is born, and this calf has come from the somatic cell, which we have used as a donor cell, and it will have the characteristics, the genetic information of the animal from which we had taken the somatic cell. It will not have any information from the ovaries or the oocytes, or it will not have any information from the animal in which womb it remained for gestation period. This mother is only the foster mother, and genetic information comes only from the somatic cell. Okay. So some facts about the animal cloning. Normally, some people have some confusion, but here I want to clarify the facts that clones are not genetically engineered animal. These are only asexually produced animals, and the genetic information of the animal is not changed. We are not inserting any DNA from other animal or species. So offspring of the clones are not clones, because when we clone the animals, it may be male or female. If we clone male, then its bull will produce the semen, and that can be used for artificial insemination. And if we produce female, then it will be like the other females. Aging is normal in clones. Clones and their offspring products are safe for human consumption. That was FDA in 2008. Labeling is not required for cloned animal products. There is no indication that clones or their progeny would pose additional environmental risk. And many countries are using this to improve the quality of the animals for food production. Then comes that, can there be any step which may be associated with risk? So in the risk analysis study, if you see, we use the oocyte, we use the donor cell for cloning process. So there is no risk in this. Till now, the pregnancy rate is low, and that is now being improved. And health risk to the animal is same as IVF, and as risk to progeny, there is no risk. So then the animal cloning companies, because in other countries, the research on animal cloning is being taken up by companies. So these are different companies, their country, and animals being cloned. You can have a look on these: the dog, cat, horse, equine, bovine, caprine, camel, pig. These are the animals which are being cloned by different companies in different countries. In India, though cattle and buffalo both have been cloned, recently a cow clone has also been produced, but main focus still now remained on the buffalo, and main focus is to produce the elite bulls, improvement in the efficiency, conservation, genomic and epigenetic studies, and health and performance of the clones. So why we clone the buffalo bulls? If you see, in 2017-18, the semen dose produced were 32 million for buffalo only. And in 2018-19, so by 24-25, we will require 50 to 60 million semen doses. And we do have bulls, but we don't have the quality bulls or the very superior bulls, which are really very useful for the genetic improvement of the animals. So these are the available clones in the country with date of birth. You can have a look on the, now these clones have grown as adult and have been trained for semen production. These bulls are available at CIRB. These are also available at CIRB. This one is a SME buffalo clone. So this was produced from the somatic cell from the SMS buffalo, the cells were collected from Assam. The tissue was brought to CIRB, cultured the cells, and then the cloned embryo was transferred 100 kilometers away from Hisar in field dairy, and we produced this cloned animal. And after 1 year, it was brought to CIRB. Now it is in semen production, and we have 4,000 doses of this bull. From rest of the bulls, we have 15,000 semen doses, and we have used some 5,000 semen doses till now. These are the cloned bulls available at NDRI. These are also cloned bulls available at NDRI. So major achievements. The 7 clones from a single breeding bull M29. This has been recorded in India Book of Records and Jimca Book of Records. 3 clones from another bull, 2 clones from bull MU2501. Recloned bull, female clones have also been produced, and pregnancies are also existing. And this is our CIRB first clone bull. Now it is 9 years old, and these 2 are the progenies of Hisar Gaurav, the clone bull. These have also become adult and producing semen. The semen of these bulls have been analyzed, and they are found excellent and equal to other breeding bulls. This SMH buffalo bull has also grown. You can have it look. And these are the young bulls of M29 and recloning. And we have produced several progenies using the semen from the clone bulls, both in the institute as well as in organized dairy farms and in field also. So you can have a look on all these calves are produced from the semen of this bull, clone bull. Similarly, at NDRI Karnal also, these clone bulls were used for semen production, and these are the cows born at NDRI Karnal. So you can have a look that more than 2 dozen clone semen's progeny have been produced in the institutes and then in field also. We studied the fertility of the bulls, and fertility of the clone bulls was similar to the non-clone bulls, both in artificial insemination as well as in IVF-based study, and this has been published. Also, we studied long-term 5-year study on clone bull cellular physiology, cell cycle, embryo development, epigenetics of the cells, gene expression, pregnancy rate, health of the clone bull, fertility, and we concluded that growth, health, and fertility of the clone bull is similar to non-clone bulls. This paper was published in Scientific Reports. It is 5 years research result are published in this. We do the biochemistry of the clone bull and the telomere length of the clone. Biochemistry was similar to the normal breeding animal, and telomere length, those who understand the cell division process, if we culture the cells, after certain time, suppose in somatic cells, after 50 to 60%, the cell stops growing, even if we provide the best culture environment to them. But this doesn't happen in stem cells. Stem cells have infinite potential to multiply. But somatic cells and body cells, they grow and multiply up to a limited period. Reason is this, that DNA methylation and telomere length shortening are the two processes which leads to cell senescence. Cell senescence is an inbuilt mechanism in any cell that the cell will grow for a finite period, and during this process the telomere length gets shortened. When it reaches at a point, then further cell division stops. So we compare the telomere length of the clones with the age-matched AI-born animals at birth at 1 year, 2 years, and we found that the telomere length was either normal or elongated compared to the age-matched controls. This gives us information that the lifespan of the clones will remain normal like the other animals. Spermatozoa global transcriptome analysis of the clone buffalo bull were also studied. In this, the mRNA and miRNA regulates the spermatogenesis, bull fertility, and early embryonic development. And the NGS data reveals that the expression level of mRNA in spermatozoa of clone bulls and their somatic cell donors that regulate spermatogenesis, fertility, and early embryonic development are non-significant. There is no difference was found. Similarly, the NGS data reveals that the expression level of miRNA was also non-significant. So, till now, we have a accredited semen lab with FACS and CASA facility. And this is the only ICR institutions lab which have accreditation. That means we can share our semen as per guidelines of Government of India. 13 clones of superior breeding bulls and 1 elite female have been produced. Adult all clones are in semen production. Now, 2 years back, DBT has approved another project to us to use the clone bull semen for artificial insemination in an aspirational district. So we have 25,000 somatic cell bank of elite buffaloes, both male and female and clones. More than 60 animal cells have been stored. Male clone progenies are in semen production. Female progenies of clone bull are also producing more milk than their mothers. And we have improved the cloning efficiency from 1 to 6 to 8%. And fertility of clone bulls have been studied. So this is about the DBT project I was talking, that we have done till now. We have done more than 3,000 artificial insemination. This is the figure up to January 2023. But till now we have 3,300 artificial inseminations, and we have 45% conception rate. So this way, with a larger data, we can say that the cloning, the females and males produced through cloning, and the conception rate and the reproduction and production of the clone males and females and clone males progenies and female clones progenies are doing as normal as normal bred animals. So this is some different lactation for scientific study. And these are the cows which have been born in field using the cloned bull semen. Until now, more than 500 cows have been produced, and many more are expected in coming months. And this work has been covered very well in important newspapers, TV channels. In Hindu, the report was published. This is in Hindi paper. And this clone I had talked, that Assamese buffalo clone, you can have a look that when it was small, young, with its mother was Murrah, you can see the black color, and this calf born from this animal was brown in color. And this was published on the cover page of Current Science. That means journal has given quite good importance to this research that they have covered or published the photo of this calf on the cover page. Also, you can visit the Rajya Sabha TV and put the words SME Buffalo Clone on YouTube. There is a 4 minutes video which will further explain about the process. Then comes, some people say that what is there any ethical issue also concerned with? So we have searched the literature and with our own experience also, there is no point of any pain or suffering to the animals during cloning, as well as products from the cloned animals are safe for human consumption. And important thing is we need to develop the societal consensus so that people should be scientifically become aware about the cloning science with the data. But we can't ignore the public sentiments, so we should first make the people and the stakeholders well aware about the science and then move ahead for this. So this is our team members, our ex-DG Saab, DDG Saab, and our cloning team members. So now it is open for your queries. If you have any questions in your mind, you are welcome.
Well, we just participants to clarify their thoughts or their experience from the KR. Do you have any questions from participants?
Okay, if you don't have questions, then I will tell a little more about the animals which are used as recipients. When we produce the cloned embryos, we have to transfer these in the foster mother. They should be synchronized. So we do ultrasonography regularly and see the animal is cyclic. If it has corpus luteum, then we synchronize it as per our need and then transfer the embryo. Main thing is this, that we should know about the reproduction of the animal, and in which, if we are clearly observing the estrus and then corpus luteum and transferring the embryos in the same site where there is corpus luteum, there remains more chances of conception. Also, at regular intervals after pregnancy diagnosis, we check through ultrasonograph after 1 month, 2 months, 3 months, because there remains some pregnancy losses. And these may be more in cloned embryo transfer. But we also did the ultrasonography in normal herd, and we could find that the embryo losses which are taking place from 28 days onwards, there are more losses up to 60 days, but even after 4-5 months of pregnancy, there can be losses. Early losses are, we can't detect the fetus, but if this fetus has grown, then this can be. And one more important thing is that we have to take care of the calf, because this becomes very important. So we try to provide the good environment to the growing young ones, and feeding of colostrum and then deworming, etc., so that we can have well-grown cow. So with all this, I thank you very much. If you don't have questions,
So this is one query from participant. We asked what's the difference between CT and cloning, and BP experience among them. And second part is what are the basic instrumental instruments for cloning experiment?
instruments.
Instruments, and we asked what are the difference between
Okay.
CT and cloning process.
For cloning lab, we require zoom stereo microscope, IVF workstation or laminar, CO2 incubators. We require minimum 2 incubators, because for cell culture and for embryo culture, we can have little difference in the temperature, and we need not to open the incubator very frequently in which the embryos are cultured. But the most important machine which we require, if it is through micromanipulators, then we should have a micromanipulator. Otherwise, we should have a electrofusion machine. Electrofusion machine gives very small current in millimoles, so that the somatic cell and the matured oocyte cytoplasm gets fueled and becomes a rounded structure to grow in an embryo. If you are using the micromanipulator, then micromanipulator is having an inverted microscope attached with the facility to handle the oocyte and cells. And looking in the microscope, using the hydraulically controlled jaws, you can enucleate the oocyte. In this case, in micromanipulation, first the matured oocyte is stained with HEX stain, and under the fluorescent microscope, we have to pick out the nucleus of the oocyte. But in handmade cloning, we surgically cut the zona removed oocytes part which is having the nucleus. So this is the difference. So the main machines are either micromanipulator or electrofusion machine, CO2 incubator, microscopes, laminar, and cell storage facility, and rest small things which you require water purifier and other things in the laboratory. As far as in the farm you need to have an ultrasonography machine and then the AI gun, artificial insemination gun, and sheath and other protective things so that we can transfer the embryo without any infection hygienically. So these are the main machines, but you require many recipients because the success rate is not that much. So in every cycle, if you produce embryo, when we culture the embryos, we get about 10 blastocysts, 15 blastocysts, 6 blastocysts. So for transferring this much number of blastocysts, you need to have several recipients. And once you have transferred the embryos, for 30 days, these animals are blocked. So next week, if we have embryos, we require another set of animals. So this way, if we transfer in 4 weeks about 6 or 7 animals, so about 30 animals, 40 animals will be required. And because all animals don't remain in cyclicity all the time, so a separate herd of recipients is required for doing this type of research or production of the animals. Okay, anything else?
So I would like to supplement that this procedure is initially very simple, but it is indeed very difficult. And only a selected level we are working on this technology.
I could not understand because the voice is not clear.
Okay, I'm saying that this technology seems to be simpler, but in fact it is complicated.
Right, right. It is complicated,
And that is
but
why only a few levels we are working on this
yeah,
technology.
it requires a huge funds, a very dedicated team, and the team must have both laboratory scientists who know the embryo culture and cell culture, molecular biology, and all modern biotechnology techniques. And we need an expert who should be very good in handling the animals, transferring the embryos, because when we transfer the embryos, the animal is not in heat, so its cervix is closed. So to transfer the embryos in the closed cervix definitely requires very good skills in the person who is doing this job. And then ultrasound knowledge to check the pregnancy. So it is a huge teamwork. Then rearing of the animals, then semen collection, semen freezing. So it becomes a big exercise, and several experts and team members with team spirit and good behavior of the people so that they can work together. This is must, and it becomes very rare. That's why many institutions are not taking this type of work. Only money can't do. You require definitely many other things.
If there is no further questions, we can stop here. So I thank you, Dr. Yadav, for your valuable presentation. And we must hope some of the team will get some points out of that. Excellent start here. Research work with or related to it. Thank you, sir, so much.
Thank you all. Thank you, Dr. Bilwar Singh. I am looking your face. Rest of the friends, I am not aware who are listening, but my good wishes to all of you. Thank you very much. Sonu,
Hello. If you have any questions, please ask.